Howdy, here is a quick benchling tutorial, mostly to show people curious how I build these terribly monstrous BSA-1 plasmids, but at the end it'll also throw in a little bit on how to clone some new fragments into them, like we will be doing at the beginning of the iGEM season. So, first of all, this is PET21A. It is a common expression vector that belongs to the PET system of expression vectors. They all have There's a bunch of different numbers of them. They share certain, properties, but they might change their antibiotic resistances between the numbers, and also the structure of this multiple cloning site here. So of interest for us, because we want to eventually express a protein and then purify it, we need something to tag our protein that we're going to express so that we can separate it from everything else in the cell. And that, in this case, is a 6-his tag. So this vector already has that built into the backbone here. It has an optional T7 tag at the front, but we'll be ignoring that when we clone things in. And then up here is, of course, the promoter, so this is where transcription will begin. The lac operator for controlling it and making it IPTG inducible. And then here's the ribosome binding site, so then translation will start from here onwards, from this NDE1 site, specifically that ATG in the sequence right there. Cool, this is a great vector, fantastic, lovely, but if we want to clone things into it, these vectors are still designed with classic restriction cloning in mind. So what that means is we'd have to pick two of these enzymes, the ones that have been picked by the people before us, and so I'm using the same sites. These are the plasmid that we got from Andy Hitchcock, one of our supervisors, and Ian Lidbury. They made a plasmid that inserts a gene, the PAF-A gene, which we're going to express and purify, between this NDE1 site and then this XHO1 site here at the end. So for our purposes, we want to insert all of our genes into this exact same region of the plasmid. We want the same spacing between the RBS and this ATG start codon, and we also want to make sure that at the end of our protein sequence we get this 6-his tag expressed. But what we want to do is, instead of using these two separate restriction enzymes, we want to use just one. We want to do type 2S assembly with just the BSA1 enzyme, which can cut outside of its recognition site and generate different overhangs. So in the end, you'll just need to take this plasmid that we make and we prepared, this backbone plasmid, and you just put it together with the PCR product of your insert. You incubate them both with BSA1 and ligase, and you just transform that product. There's no gel purification step, there's no separation of sizes, you don't have to use two different enzymes or do enzyme inactivations. It keeps everything one step, which should make cloning hopefully very easy this summer. But complex part, of course, is how we actually make that vector. So, starting from this backbone, there are a couple of things to realize. One, I guess a couple of things to think about as goals. One, we want to replace this site between the NDE1 and XHO1. All of this sequence we don't need or care about. We want to chop it out and replace it with eventually our gene. Or in this case, as we're building the backbone, the BSA1 sites that will replace all these other restriction enzyme sites. There is, however, one very annoying problem with this plasmid, which if you look over here on the map, where you can see certain restriction enzymes, I've highlighted, I'm looking specifically for BSA1. There is an existing BSA1 site right over here in the middle of this ampicillin resistance cassette, which is infuriating. So the little secret magic trick I'm going to use here is I'm going to change one of these codons so that this BSA1 site, which you can see there highlighted in blue, is no longer present in the ampicillin resistance cassette, but it still produces the exact same protein in the end. So when and in fact I guess I can show this now, to see what protein is being produced, and this is for some reason in two parts, this annotation, I do not know why. If you want to see what protein gets produced in Benchling, you can highlight a set of sequence, you can right-click, and then go to Create Translation. Here we want a forward translation, so it's on the top strand. You can tell that's the direction we want, because that's the direction our AmpR annotation is going. So I can click that, ignore all these settings for now, and just save. It'll pop up a new translation here, where you can see now the amino acid codes for the final produced protein. And you can see that's a stop code on there, and of course we have our, we should of course have our ATG. Oh, fascinating. So I misclicked that. Okay, you get to see some sausage factory stuff, so I messed that up. So, what I must have done there is when I clicked on this second amp bar, I must not have held down shift to select both of these ampicillin resistance regions. So if you want to get rid of a translation, you can right click on it and delete it, and then we can do the same thing here. Again, I'm not entirely sure why these are two separate annotations. Yes, okay, so that's the problem. If you right click on the annotation itself, it'll reset your selection. So if you select both of them and then right click above on just the sequence, it won't reset it. Right click on the annotation and it resets it. That's a Benchling quirk to be aware of. So right click on the sequence and then do the same process as before, creating a forward translation with the default settings. Now we can see that it starts with a start codon, ATG, methionine, and then we go along, have the whole protein, and we have a stop codon here at the end. Now, the trick that I've played here is I've designed some primers that are going to replace one letter in this sequence. So currently, you can see this codon here for this glycine is a GGG codon, so 3 G's in a row. What you can change this to, but that means the same thing as a GGC codon. The reason this works, ignore my spelling. Right, so the reason this works is that you have a lot of redundancy in certain codons. So the way you read this particular chart is from the inside out. So say we have our GGG, you can see that leads us to this glycine, a GG and a G glycine. But also a GG and then any of the other bases. Note here that U is just the RNA equivalent of T, so replace U in your head with T here. But we could put any of these other bases in that third position and it wouldn't change it from a glycine. So what I've done is I have changed this last G here into a C. So you can see up in that primer that we're going to be using to rebuild this vector out of these two pieces. I've changed it to a C, and down here I've changed it to a G, where on the antisense strand it used to be this C. So I've just swapped a G for a C there. That single base change will get rid of this BSA1 site, because you can see it disrupts that recognition sequence in blue, but it will keep the coding protein exactly the same as before, the same exact glycine amino acid residue. Cool, so I'll get rid of that translation I created. And then move on to explaining the next part. So the point of this, of cloning this in two halves, splitting it, and then inserting that C, of course, is to get rid of this BSA1 site. And then there's the second half of the cloning we want to do, which is replace the multiple cloning site. So if I jump back over there, we can see I have some primers here. Now, there's two sets of primers. That's a bit of a spoiler for later, because I'm both going to be reassembling this plasmid as is, like this, with the BSA1 sites, but then I'm also going to be creating a version that has some green fluorescent protein in between the two BSA restriction sites, so you can tell if your assembly has succeeded when your colonies lose the green color. It just helps us for early screening and saves you from having to do as many colony PCRs down the line. So, the ones we're interested in now are these non-GFEP ones. So this one up here, this top longer one, the NTS Rev, and then also this one down here. Now, the way all of this assembly is being done, because we have problems with restriction sites all over the place, if we wanted to build this with Golden Gate, so that's using these type 2S restriction enzymes, we can't use BSA1 because we're trying to insert BSA1 sites into our final product. What we could use is the alternative that's very, it's a very efficient enzyme, it's a nice one to use, which is BSMB1, but there's a problem with that, which I did not find until designing the entire construct. BSMB1 also already has sites in this pet vector. One in this LAC-I gene, and then another one of these sites down here, in the middle of the plasmid backbone. So, we also can't use BSMB1 to do this cloning, because we'd end up digesting this backbone piece, which we want to stay, in one piece. So the final assembly construct or strategy we've gone with is actually Gibson assembly, sometimes called hi-fi assembly, but the basic idea is these primers are going to add some homology. So the new sequence being added by these primers, and you can tell it's the new sequence once we lose the colored region. It goes on to this, the uncolored region over here. That's the overhang. It's a new sequence we're adding. All of that is going to then match up with whatever it's getting assembled to. So it'll act as if it like base pairs directly. Now, to see what I mean, we can take a look at one of the PCR products. The way I designed this is a bit out of the scope for this particular video. There are ways, it's important to know, to use this assembly wizard down here and select this Golden Gate homology-based cloning. And there's even a new assembly tool available to do the same thing. But the point is, this is a tool that will help you automatically design these sorts of primers if you're just trying to stick two pieces of DNA together. I'm trying to do something a little bit squirrely, which is stick two pieces of DNA together, but also in between them add new sequence. Because I need to introduce, as we'll see in a moment, these BSA1 sites, and then also the sequence that they're going to cut. Because I'm inserting additional sequence that kind of makes it difficult to use Benchling's existing assembly tools, at least as far as I'm aware. So these particular overhangs I designed by hand, but I will be showing you how I verify that I've designed things correctly, which is a crucially important step because it's very easy to get these things wrong. So enough of that preamble. Let's take a look at what happens when you create a PCR product, or two of them in fact, out of some of these primers. So if you notice the primer directions, these are actually going backwards, away from this multiple cloning site. If you want to insert new sequence, you always have to do that on the 5' end of a primer, the beginning part, because the 3' end always needs to run into the rest of the sequence because of the way that DNA polymerases work, and the fact that they always run from this 5' to 3' end. So, this primer here actually goes back into the backbone up this way, and will eventually meet this primer here, which goes in the forward direction. So these two primers make a pair, and you can see that that covers most of this backbone here, all the way up until this ampicillin resistance site. Again, the reason we have to cut it at the ampicillin resistance site is because inserting any new sequence or changing any sequence requires that you put something at the 5' ends. So we need to get those 5' ends by creating two PCR products here. So that's why we have a cut to get rid of that BSA1 site. And then likewise, you have now this primer going reverse direction from here, which will then end up at the opposite primer that goes forward from the MCS, which is this one. And so you can see this gets that other section of the plasmid and copies that up, adding the little overhangs along the way. So if you shift-click two primers, like I just did there, and you right-click in between those primers, you get this option to create a primer pair. Now, that would probably be where it currently says show primer pair, because I've already linked these two primers. But the point is, this will then give you some additional information about the melting temperature of these two things. You want the temperature difference to be as small as possible. You can take a look at some secondary structure to see if they fold in on each other. But what's relevant for us is this create PCR product button. So I have, in this particular case, already created the PCR products. I'll show you in a second how to do that properly with our second example. But first one, here I've created some PCR products for those two different fragments we just talked about. Here is the longer fragment containing most of that backbone, this LAC-I region, the origin, and part of the AMP-R, the end part. And then here is that smaller insert region that contains the rest of the ampicillin resistance gene and that his tag and everything that's in the multiple cloning site. So this PCR product corresponds to roughly this region, and then this longer PCR product corresponds to very roughly this region around here. Yeah, around there, we'll say. So, how now do we take these two PCR products and assemble them together? Well, first it's worth looking at, just a sanity check, that we have indeed, 1, inserted homology, and 2, inserted BSA1 sites where we wanted to. Now, okay, quick more tangent. Not really tangent, but on the fly website pulling up, taking its time because it's the iGEM website. Thank you, iGEM. Okay, and then zooming that in so everyone can actually see. The way type 2S enzymes work, as you can see here, when I highlight or roll over one of those enzymes, the blue region is the region of DNA that is recognized by the enzyme. So wherever that blue sequence is present, there's going to be a cut in the DNA. Normally, with non-type 2S enzymes, that cut is inside the recognition sequence like this. So the blue box and that, cut line, this diagonal, well, I guess, yeah, 90-degree cut line that goes through, those will overlap. Here, you can see they don't. So the recognition site is outside, and it cuts downstream over here. Now, you can imagine if the recognition site and cut site overlap with each other, you'll always get the same overhang. So for example here, if you digest with AGE1, you're always going to get on the top strand this CCGG sequence all the time. You have no other option, because if you didn't have a CCGG there, it wouldn't be cut at all because it wouldn't be recognized by this enzyme. Here, we can recognize this sequence that's upstream. So this sequence, as long as this sequence is there, it'll cut whatever sequences down here. And this we can change because it's no longer inside of this recognition site. This is the secret with this type 2S cloning. What makes it different from this other restriction cloning is that you can generate two different overhangs from the same enzyme. And then the reason this is important is that you can make sure that your gene gets inserted into the sequence in the correct direction. So there's like a front and back of the sequence. So here, in this case, I've decided to steal iGEM's existing standard for this. So iGEM has its own type 2S cloning standard that's a little bit complex, and there's some more nuance to it. You might be familiar with it from your interviews when we looked at the Marburg collection, which is another version of kind of the same idea. But what is important for us is that iGEM does standardize these overhang sites for coding sequences. So here we can see, if we want to put it in a coding sequence, a CDS, so that's like, a protein to eventually express, it always starts with an overhang of AATG and ends with an overhang of GCTT, or, CGAA, depending on which strand you're reading. But sense strand, AATG, ends with GCTT. So what I have done is design these BSA1 sites using the primer sequence that's been inserted. So you can see everything here. This is all newly inserted primer sequence. Here is, of course, that AATG overhang, and down here is that GCTT overhang. And both of those are being generated by BSA1 sites that are in the middle. So this BSA1 site is there, up to this CC, and then this BSA1 site starts immediately after that. So that is our new cloning strategy, and we'll get to see in a little bit how that actually works. But in the meantime, let's actually assemble a final plasmid out of these two pieces. If you want to convince yourself that we do indeed have Gibson homology before we use the tool to automatically find it, you can do something like, I don't know, copy this sequence up to here, using controller command C, and then jump over here, and then controller command F to search, and paste in that sequence and look. Here we have the exact same bases as we do over here, so that's that homology that's going to help stick these things together. And then everything upstream here is also homology, but that just is going to bind farther back. So instead of from there onwards, if we search, now it starts from there back. And so point is, these two bits of DNA will overlap, they'll base pair with each other, and then the Gibson assembly can then stitch them together. So let's actually do that Gibson assembly. In fact, I've already done this, so here's one I baked earlier. I'll show you again in a second doing this from scratch. There's a number of ways to actually go about this. You can use this assembly, the assembly wizard, and then go to Gibson. This gives you one type of tool, which you can see down here. But I'll just go this assembly for now. But there's a new tool that Benchling has introduced that's much more sophisticated. So if you're doing very simple things, it might be more than you need. But it is a nice way to assemble things, a nice alternative way to assemble things that gives you more control over what's actually happening. So you can get to that by doing this plus and then assembly, and then assemble sequences by cloning. And we'll see that whole process in a little bit, but point is, here is the final one I've created for now. Here you have two different fragments. You have this backbone sequence, which I've set arbitrarily as this longer PCR product, and then we're inserting this shorter PCR product back into it. You can see here that we are, well, you can certainly see somewhere that we're using Gibson assembly. Yes, up here at the very top you can see Gibson assembly, so that's the type of assembly that's being done here. And you can also see under this gear some different details about that assembly. So here the homology has to be at least 20 base pairs, but no more than 30, and the TM of that region has to be at least 50 degrees. So there are some parameters that we'll get to set when we initially create this. Those are the defaults. So basically, after you pick these two different fragments, there's a button here that'll say auto-populate, which we'll again see in a little bit, and it'll automatically generate a final construct for you. In fact, we can see here. So here it shows us those are the regions of homology. There's our insert, and there's our backbone. And if we click on this sequence, it'll show us the proper map that gets generated. So here we are, our final plasmid, exactly the same as our original PET21A, except in here, there's been a tiny single base change, which we should be able to see because it's been put in lowercase, but maybe that's too brave of me to think that I can find a lowercase letter in a massive ocean of DNA. I think that is indeed too brave, but rest assured there's no longer a BSA1 site there, because you can tell when we highlight our BSA1 sites, it says there's only 2 of them, and both of those sites are in fact, have I just deselected that? No, I've not. Okay. Both of those sites are in fact over here in the cloning region. There's 1, there's 2. So we've gotten rid of the restriction site in the ampicillin region, and we have these 2 new things here for generating overhangs. Fantastic, job well done, great. Now I'll quickly show this exact same process, but now inserting this green fluorescent protein into the middle, and then I'll finally round things out by showing how to actually clone something into one of these vectors. So this was the initial version that we created as a proof of concept, and then Mario, one of our lovely advisors from last year's iGEM team, suggested that we actually, since we're recreating so much of this plasmid anyways, why don't we also throw in something that makes selection a little bit easier? And so that's why we've got this green fluorescent protein in there. The idea is, during type 2S assembly, or Golden Gate cloning, whichever you want to call it, sometimes you will end up with original plasmid that you put in, so this original backbone that does not get digested. Now, when you transform these into semicoli and put them on a plate, they will grow on ampicillin antibiotics, so they'll look like correctly cloned colonies, but in fact you'll still have this empty cloning site up here, and there won't actually be a protein inside. You can run a colony PCR of this region and look at the size of the fragment generated, so you can make sure that you actually inserted some sequence. But the colony PCR process can be a little tedious, and it can take quite a while to get results, whereas if we generated a version of this construct that in between these 2 BSA1 sites here had a green fluorescent protein that was expressed, then colonies that did not get digested, so ones that are just like this, intact, will glow green, and so you'll just know to pick the non-green colonies, because that means something has replaced this original sequence in the middle. And it's a very useful strategy to use. It's kind of like a more modernized version, I suppose, of blue-white screening, if you're familiar with that. So, moving on now to a second version of this plasmid that has GFP thrown in it, specifically this SF-GFP. I'll go ahead and clear out our workspace, close our tabs, and we're going to do this one from scratch together. We're going to have a great time. So, step 1 is, again, to hide the pre-existing baked cake from you. So I've already, in fact, made these PCR products. Yeah, and in fact, because this is boring and I'm just creating folders and moving things around, I'll show you how to do it exactly once, then I'll pause the recording and come back. So, like with the last one, I had a folder called PCR products that I created inside to store all of these so that when I'm looking at these different folders, I know what DNA is what DNA. So here's something that you might not know about Benchling. Here, in this current compact list view, there's no way to select multiple files at once. So if you want to move these to PCR products, for example, you have to do right-click and then move to folder and do this one at a time into the PCR products folder. That's horrible. Don't do that. Don't put yourself through that. So instead, you can use this little arrow up here that opens up this file thing into like a full-size file browser. It gives you can see a little bit more information sometimes, but also, importantly, allows you to select multiple files at once and do bulk actions with them. So you can archive them all at the same time, or in this case, move all of them to a folder. So I can move both of those at the same time into the PCR products folder. So there you are. There's a little Benchling insight. Now I will do the same, but this time copying some sequences to create that template folder, and I'll be right back. Alright, welcome back, my dear friends. I spent a little bit of time just up there copying a couple things into this templates folder. So if you'll recall from before, again, we have our same pet21a. This one hasn't changed from before. It's the exact same sequence with the exact same primers attached as we previously had. So nothing new or wild here. What is new and arguably wild is our pjump 28A 1A plasmid. This is one that comes in the iGEM cloning kit, and this is one that you may be very familiar with if you are familiar with the Marburg collection paper, because this is a similar style. Right, sorry for the pause. It probably sounded like I was having a stroke there. I had to double check that I wasn't insane, and that you actually did read the Marburg collection paper, and not this separate jump paper. Rest assured, it's the same sort of idea with the cloning, with these modular overhangs. This is just one example. This is a slightly different system that iGEM ships with all of their distribution kits. So, point is, it's a plasmid we had on hand that importantly has this super folder GFP gene that makes the cells super green when it's expressed. So, what we want to do is take advantage of these BSA1 sites that it's already got here, clone out this GFP region, and then insert that into that multiple cloning region of our new plasmid. And so the idea is once these BSA1 sites are then digested, they leave the same overhangs that we want. You can see me here adding in that new overhang, the CGAA1. So we're adding those new overhangs, but we're keeping this reporter gene in the middle. Basically, everything else is exactly the same as we previously looked at, except we're now going to be creating those PCR products with these new and different primers here. So instead of the longer ones we used before, we're going to down here use these ones that have the GFP in their name. The reason this region of homology here is a little bit shorter is because we now no longer need to add this BSA1 site manually, as we were doing here, at the end of this sequence, because that BSA1 site is actually present already in our template, that pjump template, so we're able to amplify that using the PCR reaction instead of adding it new on the primers. So, same process as before. You would go through and create your PCR products by pairing these different primers and then going to your linked primers and then doing, so if you go to primers and then primer pairs, you can then select any of these ones and then create a PCR product, and that's going to produce the files that we're looking at in just a second. So again, these ones have already been produced in this particular example. I will show you just so you can see why not. So here's one that has the green fluorescent protein. So if you want to look at, say, secondary structure. So will these primers cause me problems during PCR? The answer with these crazily designed primers is probably. This is really bad secondary structure, which is why I'm doing this cloning instead of putting it all on you guys, using a nice polymerase that we have in our lab, and my willpower will be the thing to take the psychic damage instead of all of yours. So theoretically, when you're designing primers, you have things that don't fold in on themselves just this much. You have a much closer number to 0 for the structure energy. But in this particular case, it's kind of what we're stuck with since we're using a really long region that already folds in on itself, and also we need to add this homology. So our hands are a little bit tied here, but the point is, here you can check what happens when two of one primer come together, when one primer itself just folds, or when these two different primers in the pair come together in combination. And you want all these energy numbers to be as close to 0 as possible. 10 is pretty bad. I would try and keep things under 6. That's just an example of what we're currently at. We'll see how it goes. Point is, from here you can then use this create PCR product button. I leave all these things at default here. This is just asking what you want to copy over, and if you want to change some settings or keep some information. Defaults have always been all right. You can then do create, and then I'll throw it in the PCR products folder. Here we are, and I'll go navigate to that PCR products folder, and I'll probably see two of the exact same thing. Yes, so I guess two very similar things. Here it looks like I've renamed the original plasmid from PET28A to PET28A+, because that's its more proper name. But the idea, or the point is, these two sequences here are identical. So this is the one I created earlier, and here's the one I created just now. Both in the exact same length 3908. And then for the rest of them, I won't show, creating another PCR product. We've already done that once. So what I'll do is I'll just archive this one that we just created now. And just for the sake of consistency, I'll rename these last couple to have the plus in their name, just so they're up to date with the template. There we are. Exhilarating stuff. Okay. The other thing, of course, that is different from before is before we only had 2 fragments, but now we have 3, and that's because we have this third piece here, which you can see is just carrying that GFP gene and then our BSA1 site. So this is what's going to go in the middle of that multiple cloning site region from before. So let's stick all of those together. Jumping back here to this folder, we can now do what I promised I would do earlier. Let's close a couple of things and only leave open the things we're going to be assembling. We'll see why that's helpful in a moment. So I'll close that template there and close this template here, keeping just our 4 or 3 PCR products. I'll go to New, and I'll go to Assembly, and I'll assemble DNA sequences by cloning. Here, we get a couple of different options. You get homology, which is a very, loose kind of assembly thing. It doesn't impose many restrictions on you. This might be useful if you're trying to maybe delete a region in, say, a bacterial chromosome or something, where you have like 1 kilobase of homology. So this just lets you kind of do whatever you like in terms of complementary overlaps that stick things together. For us, we can use Gibson, which also assembles things with homology, but it has a couple of extra settings that let us either generate the overhangs and this homology with new PCR primers, or in our case, since I had to design these primers by hand to insert that sequence, I'm going to have to find existing overlaps, and so that changes our settings a little bit. I can leave this at default for this 20-30 base pair range overlap, and same with the temperature. Here we need to turn fragment bends from 2 up to 3, because there's 3 pieces we're going to be assembling. The final plasmid will be circular. We can put it location-wise in this parent folder, not necessarily in the sub one. And the assembly I can call the name of this construct, so that's going to be PET21A plus, and then the BSA1. Now it has SFGFP in as a reporter gene. Save. Fantastic. So, here, theoretically, it will now show up. Yes, fantastic. So there we are. And we can see our same assembly tool from before on Gibson. Still using legacy stuff? No, I'm not. So, there's 3 bins. One's at this backbone, and then there's 2 different inserts. In our particular case, it doesn't really matter, backbone versus insert. They'll all be the same, I guess, for us, because we're building a new backbone. I think the idea of this backbone versus insert thing is that the backbone is what is assuming is in every construct because the antibiotic resistance, but I've not used it for the combinatorial assembly too much, so I could be wrong on that one. Point is, for what we're doing, which is nothing combinational, so what this does let you do is select several fragments for each of these bins and create every possible combination. We're just interested in 1 combination, so we're going to add 1 fragment for each. And here we can use this Open Sequences tab to select our open sequences we have already. So first thing we'll do is we'll select the longest PET21A backbone sequence that we have, which is this one. When you click on it, you can see it gives you a bit of a sanity check. It says, hey, do you want to start at the start of the sequence and end at the end and do it in the forward orientation, or do you want to pick like a subsequence or reverse it or do something like that? In our case, we've already designed it to be, forward direction and full length, so leave all those settings as is and go ahead and click add. Then we want to do the same for our next thing. So we have to think, in fact, I'll do this wrong just to show you what it looks like. So if I then go like before with our second PET sequence, which is this one here, I believe. Yep, so you can see this is the shorter one, this is where his tag is, etc. Toss in that one, and then finally toss in our final fragment that has the P-jump SFGFP on it. Here you can see the whole thing linearized. And I try and use this auto-populate thing to construct this final plasmid, it's going to spit back an error. Oh no, what have we done? So it's complaining about no overlaps that could be detected between the first two pet fragments. Why could that be? Well, here there's an order to these, bins and inserts, if you will. So this is assuming that directly after this backbone pet comes the next pet piece. But actually, that's not the case in what we want to build. What we want to do is have one half of the backbone, then the SFGFP that's going in that cloning site, and then on the other side, the rest of the pet backbone. So what we can do to fix this is just rearrange the bins like this. So go ahead and click on this like 6, circle thing, drag it over here into the middle, and now you can see, et voila, ready to assemble. So it's correctly detected all of our homology. So what we can do once we're done, once we have all of our constructs that we want, we can go ahead and slap the assemble button up here. In our particular case, we're fine saving the construct in the current folder, so that's great. We don't want to save the fragments here, and we also don't want to save any oligos or primers here, because, again, we're using PCR products that we've already generated using manual primers. If you wanted this tool to automatically design primers for you, this is where you'd enable or pick the location to save those. So, assemble, et voila. Again, if I jump in and out of that folder, it should now appear. There we can see this horrendously named construct. I will go ahead and save all of us the misery of looking at that and name it the same thing here as this assembly. You can still tell the two apart because they have different types. So here that little plasmid represents an assembly type, and down here that DNA helix, double helix, represents just a DNA sequence. So you can still tell the two of these apart. Here then is our final sequence for our fancy PET21 BSA SFGFP vector. So as before, the BSA1 site in the ampicillin region is gone. Fantastic, we have only 2 cut sites. Another thing, of course, those 2 cut sites are here. They're flanking this multiple cloning site, or what used to be. So they come after the ribosome binding site and the whole promoter. And the other one, of course, comes right before this 6-his tag. And then in the middle, we have our giant GFP sequence. Now this GFP sequence has its own ribosome binding site, and then hiding in here is another constitutive promoter. So that's how this GFP gets produced, and you can actually see it in all of the cells. There's a promoter here that lets E. coli express this gene, which is separate from this T7 promoter, which only certain E. coli strains can express, and is what we'll use later on to controllably produce the protein that we replace this GFP with. Of course, there's your BSA1 site, AATG, just as before. And then at the end, your GCTT, just as before. So, now, the final thing to do, and I'll pause the recording briefly just to do a little bit of preparation for this, will be to take an insert. In our particular case, we're going to want to put a faux A biobrick into this plasmid, and I'll show people how to do that. Alright, welcome back. So I've set up a couple of different things here. The most important thing that is new is this biobrick here. So this comes from the iGEM registry and a previous iGEM team who worked on enhancing the activity of this phosphatase. So this is a faux A phosphatase that they made 40 times more active, and we've copied the sequence directly from the iGEM registry, just removing the start codon for a reason that we'll see in a moment. There's a start codon built into our vector. So you can actually see 2 pairs of primers here. We are going to be just using this BSA1 set of primers. This other set of primers is for a backup assembly method if my wild PET BSA1 vector cannot be built. But what we're focused on, of course, is this BSA1 assembly for now. So again, like before, 1 primer on this end of the sequence, 1 primer on the other end of the sequence, and these are going to add an overhang. What overhang, you may ask? Well, we can take a quick look. If we jump into the primers, you can see the overhang here. So I can tell you what this is. GT-CTC, I sure hope, is a BSA1 site. So we'll double check real quick with our backbone, of course. So, bring me straight there, in fact. So, Yes. So the recognized thing, yeah, okay. A GG, TC, TC. So, welcome back, dear friends. There was another live time. That doesn't sound right when I was saying it. So I found a mistake in one of our primers. I think it'll be alright. But we'll find out, because the primers have already been ordered. If not, we'll know where to look for the issue. So, here, on this forward primer, this is a much easier one to understand. We have one base of padding, because if you have a restriction recognition site, and this is that site for BSA1, it is GGTCTC, so that 6-base sequence is the recognition site. If you have these recognition sites right up at the end of a piece of DNA, you can have trouble, because these are restriction enzymes that act as endonucleases, so they're used to cutting in the middle of DNA sequences. So if you stick them too close to a DNA terminal, then they can have some trouble actually performing their, making the cleavage that they're meant to. So that is why we have this extra T base as a spacer. Different restriction enzymes need different amounts of space. If you get your enzymes from NEB, they have this table on their website on a page called Cleavage Close to the End of DNA Fragments. So here you can find a number of different enzymes, and for our BSA1, high-5E2, the one we'll be using, whether it's one base pair, 2, 3, 4, 5, whatever, all of those work with basically 50 to 100% efficiency, without needing more than one base pair. So that's why there's one base pair of padding, to keep our primers short, but to still allow this enzyme to function fully. Now, of course, missing from this table is what happens when you have 0 base pairs of spacing, which is the mistake that I found in our reverse BSA1 site. Here you can see there's our BSA1 site, GGT, CTC again, but now it's immediately up against the 5' end. Will that cause issues? Maybe. So we'll have to find out down the line. But at least we know that that's a problem, potentially, now. Aside from that, the other thing to notice is that BSA1 will cut, and I can show you by going to its little page here, although you've seen this a couple of times already. It cuts one base pair away from its recognition site on the top, and then 5 away on the bottom. So point is, there's one base of random padding that you need before the cut gets made. So you can pick whatever base you want. In this particular case, it looks like we've picked, so there's recognition site. So there we picked G as our random base of spacing. And then here's that AATG, which we're familiar with as that iGEM standard overhang for the beginning of a coding sequence. So fantastic. That all makes sense. This is the beginning of our coding sequence, and it has that overhang. Now, if we look at the end primer, this is, of course, the one that had that problem with the spacer. So we're missing the initial spacer, but then we still have our same restriction site, 6 bases. Here's the gap of 1 base, and then we have AAGC. Now that might look confusing to you, because AAGC is not, in fact, any of these particular overhangs here. Now, the reason it looks funky is because this is a reverse primer, and so we have to read it on the bottom strand. So this primer view always shows 5' end first and 3' end second. So, but if we look at the actual sequence here where it's bound, you can see, of course, the primer is going in the reverse direction. So, looking at our assembly standards and also our primer overhang, pulling this one back up again, we have AAGC, but if we know that this primer is rotated around and it's running in reverse, then actually our overhang that gets left on this side will be the reverse of this sequence on the bottom strand, so it'll be CGAA. So if we now look here, we can indeed find on our bottom strand CGAA. Now, if this turns your head around a little bit, trying to think about which strand things are on, you're in good company. Luckily, Benchling's tools will let us assemble things and make sure that everything works out before we try and do it in the lab. So if you're ever unsure, we can check it, which is what we will do now. So I can't recall if I've already created the PCR product. I don't believe I have. Nope, I have not. So here we can jump into our primer pairs as we're used to doing. I'll pick these two BSA1 primers because I said those are the ones that we're focused on for now, and I can do create PCR product. Keep all the default settings, and then make sure I'm putting it into this new PCR products folder. So, et voila, here we are, our new part with these BSA1 sites added. So now you can see in this sequence, there's the BSA1 site. It's going to cut and generate this AATG overhang, and it has its one sequence padding, one base padding there at the very end of the sequence. Fantastic. Down here at the bottom, because we're fools, it's missing the one base padding, but that's okay because it's still going to cut and leave us with the same site. Now it's overlapping with Hindi 3, which is incredibly difficult to see. But rest assured, this BSA1 site here is going to generate this GC-TT terminal overhang, which matches what we have here, GC-TT. Now, to actually assemble these, we're going to use a different cloning strategy than we did before. In fact, I might do something wild and try something I've never tried before on camera, which is using the new assembly tool, but for classic restriction cloning. So, what we want to do now, that we have our PCR product, and then also in this folder we have our two backbones, just to demonstrate that the resulting sequence should be identical. Whether we start with the BSA1 without the GFP or with the GFP, we'll get the same product either way. So jump into the assembly wizard and assemble sequences by cloning as before. And this time I want to do not this wizard at all, which is unfortunate. No, it'll work out all right. So the reason for my brief moment of hesitancy there is because it looks like this new assembly tool does not, in fact, have the old digest and ligate option, which is the one that I'm used to using and the one that makes sense here since we've already added those restriction sites. But it looks like there might be some hope with selecting that our sites are already in there for Golden Gate. So now, of course, we're using Golden Gate cloning, we're using BSA1 as our enzyme, and we're using pre-existing cut sites. The other option, of course, is that you can use primers to add new overhangs that have these BSA1 sites. So again, if you don't want to do what I did here, and in fact this is probably what we should have done since I did not include the correct amount of padding, if you want to generate these primers on either end of the biobrick, this faux A gene, then you could say use a primer pair, and here it would recognize the different bases. And in fact here it looks like, yeah, the pre-recognition site length, 6 base pairs, that's where you would have your custom sequence that you put in for your custom overhangs and things like that. In our case, of course, existing cut sites, we've already put them in. BSA1, let's give this a go. This could be an absolute mess because I've not done it before. But basically, we can use in fact, we should open all of our sequences. So, and close the ones we're not using. So close the original one, we're keeping just the biobrick. I didn't name this assembly because I'm a fool. I'll go ahead and name it now. I'll call it Pet 21 A, and we'll give it its little plus. And then this one will be BSA1, but instead of an SFGFP in here, it's going to have faux A. Fantastic stuff. Location, not PCR products. Only a fool would put something there, so I'll move it instead to the overall cloning folder. Outstanding stuff. So I'll jump back into that folder so we can make sure, yep, there's our assembly. Great, fantastic. Now, here's where this new fancy assembly tool actually comes into play. It lets you select several pieces for each one of these, bins or these types of parts, and it will generate all the different combinations that you could generate from them. Or at least that's what I think. I've never used it for that purpose before, so let's find out together. Here, jump into the backbone, and we can open, oops, forgot to open the second sequence, like a fool. Okay, so here we have, yeah, using existing cut sites, that's what we said earlier. That's great, that's fine. We want both our BSA1 vector without the GFP inserted, and here you can see it shows us a start and an end. Let's make sure it auto-detects our restriction sites correctly. But no, here in fact it's done things incorrectly. So this is picking this site in this tiny fragment between the two BSA1 sites, when what we really want is for it to go the other way around. So if we go ahead and click this little invert button here, which is actually very useful, thanks for including that, Benchling, you can now see that it selects the backbone instead of this tiny bit in the middle. A good sanity check is always check how long the thing you're inserting is. We certainly don't have a plasmid that's 18 base pairs long. It's much more likely that it is the 5.4. And so, of course, looking at the site manually, we can see that's correct. We're keeping, for the construction, all the highlighted gray area. Outstanding. In reverse orientation, to be entirely honest, I don't know which way it's going to want to read that. I think I'm going to go with the forward orientation, as to not reverse this plasmid, because the plasmid is in the correct orientation. We want the his tag and things to be reading left to right. We can debug that later. Sick. Go ahead and add that. And now, of course, what we want to do is also add this second sequence, this GFP inserted backbone. This should be the same process. So here you can see, now it's selecting and trying to use our GFP. But again, we wanted to use the backbone, so go ahead and invert the selection and then make sure this is set to forward, and then go ahead and click add. And in fact, now, yes, you can see these have the same length, or at least they should. Yes, they do, yeah. 53366. So after you digest those, of course, the GFP is lost. These backbones should be identical. Now, of course, we want to now put in our insert sequence, which is this PCR product here. This one we don't have to do anything funky with. It auto-detects it correctly. We want the inside piece, of course, and not the tiny, tiny bits on the outside. So go ahead and add that. And we get those three pieces all put together. Go ahead and auto-populate our construct, and here we can see, two different things get generated, which makes sense, because if you, combine two pieces with one piece, there's two different things overall that you can build. So here, the insert's, of course, identical. Here, there's two different backbones, one with the GFP, one without. The overhangs are the same for both, because that's how we've designed them. And I can go ahead and assemble all of these pieces together. Ignore the schema part. Make sure that we're in the right folder, which it looks like we are. Outstanding. Skipping the DNA fragment saving, and also skipping the oligo saving, because we're not designing any new oligos here. All right, so jumping back here, there's now two new vectors. Are they, like we want them to be, identical? That's a good question. So let's go ahead and check one of them first. Let me pick my favorite. Arbitrarily, I guess I'll pick this one with the GFP, because hopefully that's the one we'll be using in the lab. And of course, I will now go ahead and rename that to be the same name as our construct file. Ta-da! Great stuff. So let's first check that this assembly is correct, and then we'll check it's the same as that other assembly, so that, whether you're using the version of the backbone that has the GFP or not, you get the same product in the end. Let's give it a go. So the most important part for us is looking at the this here transcription site and translation site, the gene that we're expressing. So by default, it's copied over this same translation from our Fo-A plasmid, but in our particular case, we're actually interested in seeing a larger translation, which is this entire coding frame, because you can see here we're missing a stop codon, and we're also, on the other side, missing a start codon, which isn't great. So what I'm going to do instead is convert this translation into a feature, so we can still keep it labeled as, this has come from the, or annotation rather, so we can keep it labeled, this is the original Fo-A. Yep, but we can then delete and recreate this translation itself. So, sick. Alrighty, where's our missing start codon? Great question. Luckily, because we've designed this well, here's our AATG site. That's the overhang, and that includes an ATG, which is a start codon. Great stuff. How far is that start codon from the ribosome binding site? 8 base pairs, just like it should be. The same distance that it is in the original pet vector. So now what we're interested in is taking that ATG, so an additional 3 bases, taking all of Fo-A, scrolling down. Also want to take all of the his tag, and now we need a stop codon, and I also know that TGA is a stop codon there at the end. So I'll select all of that sequence and go ahead and create a translation on the forward strand. Keep the default as headings, slap her on in, and here we can see our final product protein. Starts with a start codon and ATG methionine. You have our whole Pho-A inserted in the middle, and then we have a tiny little linker that comes from the annealing site, which is an alanine leucine. Both of these things are relatively unextraordinary amino acids. Alanine with a very small side chain, leucine with an intermediately large one, but both of them are just normal non-polar amino acids, so they're unlikely to disrupt the effect of the overall protein. At least that's the hope. And then, of course, following that, we have our 6 histidines in a row for the 6 his tag, and then a stop codon. So this looks perfect. It looks totally correct, just like what we want. Now the real question is this beautiful thing that we've just created actually the same? Oh, look at that. It's giving us an interesting little name there. Well, I'll go ahead and give it a better name than that. Pho-A his for the his tagged Pho-A. That'll be fine. Cool. Okay. So is this beautiful sequence that we have here of things correctly inserted with one step Golden Gate cloning into our backbone going to be the same regardless of which of the BSA backbones we start with, whether it has the GFP or not? Well, to do that, we can do a very easy check. Copy all of these bases with just a Control-C, which sometimes works, sometimes doesn't. So if it doesn't, you can go ahead and go copy and then DNA sequence. And then we want to jump over to this other construct that got built. Search the whole thing for an identical sequence, and voila, the whole thing becomes selected. If there were any mismatches in the sequence, it wouldn't highlight in yellow like this. And so there'll be other tools that you can use to find where those mismatches are, but this just checks if they're an identical match. You can see if I change one G to a C, nothing gets selected anymore. Change that one G back, there we are, everything's selected. So we can tell these two plasmids are identical, which is fantastic. So I can go ahead and get rid of this one since it's a duplicate. And we have our final construct here below. Excellent stuff. The one last useful thing, I guess, to keep in mind for when you are doing this cloning down the line. The goal is not necessarily to put in this same Pho-A every time, of course, but instead you're going to be wanting to put different mutant versions of the PAF-A phosphatase that we're working on. So we'll do our in silico redesign, we'll order some G blocks, and what you're going to want to do is then build up some new constructs out of those. So I'll do one last pause to do a little bit of thinking, and then I'll show you, I think, what will be helpful for that. Okay, welcome back to the Madhouse. Here we are going to do something I've never ever done before, again on recording, and we'll see how it goes. And I'm going to put it all in a place that I'll blow away when I'm done with it because it's a bit messy and it's not going to be useful for what you're actually doing down the line. In terms of these particular sequences, the process I think will be very useful for what you're doing down the line because it should be identical. As we go through and redesign all these PAF-A constructs, you're going to end up with a lot of versions of the same kind of gene. Now, here I've just copied and pasted the same sequence 3 times, but of course you'll have 3 different sequences because you'll have 3 different versions that you've redesigned using the AI tools. And I want to show how you might then automatically generate all of the final plasmids after cloning them into the backbone. And also, how you're going to automatically generate the proper primers that you need to amplify these G blocks up. So, you get your protein sequences, you turn them into some codon-optimized DNA sequences, and you've got 3 different versions of PAF-A here. Sick. Now, we want to put them into our same vector as we did just before, this BSA1, PET21, SFGFP thing. So, let's go ahead and create a new assembly, created by cloning. We're going to be doing Golden Gate cloning. I'm going to name it Destroy Me, because it's a temporary file. And the number of fragment bins will be 2, because we'll have 1 backbone and 1 insert for all of these things. We're going to build a final circular plasmid. Sick. Here is where we're going to want to use BSA1 as our restriction enzyme, but now, instead of using existing cut sites, we want to use a primer pair. We'll actually want to use a combination of the 2, so we'll see what that entails. Now is when I'm going to panic, because I don't know what all those options are, and instead go back to just existing cut sites, and we'll change it later, and we'll see what options it pops up. because I don't know exactly what they mean by things like the pre-recognition site bases. So we'll find out. So let's go ahead and slap that in, create a new assembly. We're going to go to our backbone, use our existing cut sites, of course, and then select our open backbone sequence. Easy peasy. You'll recognize this problem from before. Again, it's managed to select the insert which we don't want instead of the thing that we do want, which is this whole backbone. So go ahead and flip that around and then make sure the orientation is forward after doing so. Add that in. Now let's open our other path A1 sequences. So this is missing a start and end site, you can see. In fact, so we'll move back. We're not using existing cut sites here. We need to add them using PCR. So here you can say use a primer pair. Excellent stuff. So we can jump in and say add a new fragment to bin, and now select our path A1. Cool, and you can search the preferred primer as well. We'll see how that all looks in a second. Point is, remember that we'll be inserting a start codon with our overhang. So we don't actually, in this particular case, care for this ATG site. So here, just note that you can sub-select different regions if you want to only put part of a protein into a construct when you're designing these primers. So here I'll skip the ATG and I'll select the rest of the sequence, just like that. Which is a little bit interesting now that I look at it, because when I was copying that sequence I must have messed something up with the final stop codon. And because I've done that, I'll pause the recording again and see what's going on. It's just what you get when you try and be clever and you select the path A annotation on another plasmid that doesn't have it correctly boundaried. So, in fact, the correct boundary for this path A, that's the last residue here, is this K, and then these other next two amino acids are simply the linker created by this XHO1 site, which is in the plasmid that I copied it from. Point is, in our particular case, ignoring the last base. So we can actually do that in our cloning process, just as before. Jump over here. And now we want to skip the ATG and then also skip the final base there, so that we only keep the sequence in the middle and we don't add a frame shift in. Awesome stuff. Search by name for primers. Boy, I don't know what that means yet, so let's carry on. Alrighty, okay, use a primer pair. We'll see if it goes ahead and designs it. Now let's select the same things for our other couple of sequences, both for A2. In fact, just to keep things exciting and funky, let's say A2, for some random reason, skipped some initial parts of this residue. So say we've designed something, we've realized all this initial sequence of the protein is useless, so we want to test a version that, you know, maybe starts from this serine onwards. So starting from the serine onwards, dropping the last bit, we go ahead and add. And then say maybe our third one, we've decided, wow, most of this protein is junk. Let's just use a tiny fragment of the middle. Go ahead and say start on. Are there any convenient methionines? Yeah, sure, but that's an ATG. That'll get confusing. So let's just go ahead and start on vine. Yeah, we'll start there. And then just jump down, say, to, you know, here, another end of the codon. So tiny segment, that particular one, we'll go ahead and add that. The reason for demonstration purposes that I've selected 3 different ones there is because then I think it should generate some unique primers for us instead of them all being the same. Because again, these 3 files are identical in sequence because I just copy-pasted them. They won't be identical in sequence after you've redesigned them properly. And with that, I can go ahead and slap auto-populate. And this is going to go ahead and generate those overhangs. Now the nice thing about this, which is fantastic, and I'm so glad that the tool is clever enough to do this, because we're using existing cut sites in the backbone, Benchling already knows which overhangs we need to generate on these inserts. We don't have to tell it, you need these overhangs, because it knows which overhangs will properly assemble. Outstanding stuff. And you can tell it's created 3 versions because we have 1 backbone here and then 3 different fragments. Now, of course, this is useless, but just to demonstrate it, so that you can understand better how this combinational tool works, if instead of our GFP version, we also wanted to test with our non-GFP version, again, we've already checked these will generate identical constructs, but just for the giggles and to see, how this number of constructs increases, if we add 2 fragments up here, we go ahead and do this non-GFP one as well. Of course, do our normal fixing of the thing there so that it picks the right part. Yep, fantastic stuff. Go ahead and add. And now we do a clearing and auto-populate. How does that work? Outstanding, just as we wanted it to. So now, of course, if there's 2 backbones and 3 inserts, there's 6 combinations. And so you can see it generates all 6 combinations. That's just for down the line if you ever do have, say, a version of this backbone that you want to test that does not have the his tag. So you want a his tag version and a non-his tag version. This is how you could automatically generate all those combinations at once instead of doing 6 different individual cloning processes. Very cool tool. Thanks, Benchling, for adding that. Let's get rid of that rubbish I just added in. Go ahead and delete that row there. And then, yeah, okay, it'll complain because I indeed got rid of that. Go ahead and clear and regenerate the table. So here we are, 1 backbone, 3 fragments, auto-assembled. Go ahead and slap that finalize assembly button. We're going to want to put it in the same temporary folder so I can blast it away in a moment. Saving fragments is optional. Here we don't need to save the fragments. It's not as if we'll ever need to order these, but I'm going to save them just so you can see what's going on. I'll keep this in the same folder as well, just so I make a nice proper mess. And of course save the oligos. This time we do need to save the primers because we're asking it to design primers. If we want to actually order these and have them shipped to our lab, we need to know what their sequences are. So go ahead and assemble. Dun, 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 dun. Finalizing. Excellent stuff. Okay, jump back here. Let's look at the chaos we've now wrought. Oh boy, that is ugly. But point is, here are our 3 final constructs. So here is the first construct we made with Parfait 1. So this is the full-length Parfait. So in fact, you will recognize, or you should see, if we start with our start codon in the backbone, we go here to our TGA afterwards, we go ahead and create a translation on the forward strand with the default settings. We're going to get a stop codon at the end, our his tags, our nice same linker as before, the full Parfait protein, and then an M at the beginning. You can tell when this is selected, the length, 1,593 base pairs. Excellent stuff. But if you remember, we've selected, and of course, importantly, there's no double start codon here, because remember, we got rid of that, and because we got rid of that last base pair when we were designing primers as well, there's no frame shift, so the his tag is indeed 6 histidines. Let's look at path A2. So this one, remember, we picked a smaller region, we cut off some of the beginning of the sequence. So we'll still have the ATG, because that's built into our backbone. So if I go ahead and grab that, and of course go down to the same his tag, go to the TGA, create that translation, as default. Okay, excellent, we didn't knock things out of frame, so it's still a his tag there, but you will notice this one starts with SIYTG, whereas our other construct, when we had a different start site, starts with QQRPK. And it looks like our SYPTG thing is going to be somewhere else. We can find it real quickly. So say we want to know, what are you starting with, my dear friend? Where is that at in this other larger construct? We can just select this, search within it. Yeah, and we can see down here. So here's the SYTG starting region from earlier. Cool, so that's generated something unique as well. And of course, Parfait 3, this is our really microscopic one that we put in there. And as expected, here's our super small Parfait. This one, I think I might have also tried not to knock it out of frame, so we should still have our correct histidine tags, but let's double check. Boodle-doop. Yep, there's our 6 his tag just as before. But this time, this fragment is even smaller, so if I look at that Parfait fragment, I say, yo, where have you come from, my dear friend? In our full-length thing, we can see that it came from way down here in the middle of the sequence. Yeah, there you are. You can see it better on the plasmid map over there. So, cool. It's now generated 3 different unique outcomes based in the same backbone with different primers. Now, what do those primers look like? That's a good thing to check now. So, one place we can see these will be in these fragments that it generates. Now, these are very messily named, it looks like. Oh, and they don't even have the primers attached. Naughty behavior. Yeah, it looks like they have 6 base pairs, which is the standard recommended thing. So that's cool. So you saw earlier our BSA-1s had 1 base pair on the outside before the recognition site, and NEB says that should work fine. They've tested it, but NEB also says, as a blanket rule, you should leave 6 base pairs of padding. Where is that at? Here we are. 6 base pairs should be added to cleave efficiency. So that's the general rule. So that's what Benchling is doing automatically for us. Cheers, Benchling. If I use this tool, then I would not end up with a problem where I had no spacing like I did with those other primers I made. Of course, you can see there's our AATG overhang and the GCTT overhang that we're used to. This is the same process for all 3 of these different fragments. Of course, you just have different length ones. Now you might wonder, how do these fragments actually get produced? How does that work? Well, it's because of these primers. Let's see, does that attach the primers to the original template? Or is that getting too brave? Too brave as well. But that's okay, we can attach them ourselves. So, yes. Oh, you do say. Oh, it's the final construct. Well, that's polite of you, isn't it? So if we want to see these primers and where they're actually stuck on, it sticks them onto the final construct, it looks like. So if we jump over here to our path A, yes, okay, fantastic. So here's the primer, it's auto-designed for us. It's put the melting temperature at 50 degrees, which is interesting, so that's something we can customize. My lab always does 60 degrees, but you can squiggle that around. There is our AATG overhang sequence that we're used to, and then here it looks like it's added some additional sequence, which if we look at the primer more closely, of course, will be 1 thing of spacing, which is this T, and then here's our BSA-1 site, the GGT, CTT, and then here is our 6 base pairs of padding, where we previously had just 1. So you can see the nice thing about all of this, using this assembly tool in this way, is Benchling has automatically generated all of the individual primers that you need to order to do the PCR reactions on these original templates here, to then generate these fragments here, which have these BSA-1 sites, that can then be assembled into the backbone to produce these final products. And it's given us all of this in 1 assembly step, which is super sick. And with that, I think that's all I have to blabber about. I say that as if it's not been an hour and 13 minutes. Obviously, if you have any questions, I'm around, so are other people on the team. Overall, it's worth getting to know Benchling and learning how to use it properly, because it will make your life a lot easier. As is a real-life example in my case, where I did not include the correct spacing on one of my primers. The reason, of course, why I designed all those primers by hand, like I explained at the very beginning, was to insert sequence, but the 1 primer I managed to mess up was not one that was inserting sequence. That one could have been automatically designed. Cool. Thanks for watching, if you made it this far. And then have a go at these things in Benchling. This last demonstration, especially where you have these different sequences and you're using that combinational assembly to stick them all in the backbone, automatically generating primers, all of that, that will be super duper vital to the work you're doing later this summer. Best of luck, Godspeed, and see you all on Wednesday.